Priti Kari, and I'm a rising senior at Basis Sonora.
Hello, I am Blessmer Tercero, and I am an incoming sophomore at Basis Sonora.
This summer we worked in Dr. Paul Kima's lab, which focuses on gastroenterology and inflammatory bowel disease.
IBD is an umbrella term for conditions when the GI tract becomes inflamed. We are interested in IBD because over 6 million people suffer from it worldwide, and there's currently no cure.
Symptoms can be severe and even threaten the possibility of death. So our lab's main research question is, what is IBD and what are some possible therapies?
The project I focused on looked at a cellular process called pyrylation, which is a post-translational modification as a normal repair response to DNA damage. Previous research from Dr. Keela's lab found that the colon tissue of patients with IBD showed hyperpyrylation compared to the healthy tissue, but we weren't exactly sure why. Further, the lab also previously found that the hyperpyrylation occurs in cells exposed to the fecal metabolites of patients with IBD, so it must be the metabolites regulating pyrylation, but we weren't sure which ones, lipids, DNA, or proteins. So through DNA extraction, protein degradation, and lipid extraction, we could eliminate these metabolites from fecal supernatant to see how pyrylation was affected. DNA had no significant effect, whereas lipids and proteins did affect pyrylation. From here, we also wanted to check another kind of lipid, bile acids, which was previously linked to IBD in other studies. Removing bile acids from the fecal supernatant significantly affected the level of pyrylation. So we know that a combination of lipids, proteins, and bile acids must be regulating pyrylation in the gut.
My research focuses on a protein known as NHE3, or the sodium hydrogen exchanger 3. Basically, it moves hydrogen out of the colonic cell and sodium and water into the colonic cells. However, we discovered that reducing its gene expression in a short period of time enhances more cell proliferation and migration in the colon. We were able to determine this through immunofluorescence staining, where we compared the tissue of a controlled mouse with a mouse that had reduced NHE3 expression. Using different wavelengths, we were able to locate NHE3 and the cell's nuclei using this fluorescent microscope, and it helped us differentiate the amount of NHE3 and nuclei in the tissue. Following that, we sacrificed a controlled mouse and a mouse with reduced NHE3 expression as well, but we processed their colon tissue and obtained their cells, and then we cultured it. We placed this culture in a hollow monitor and let it flourish in 24 hours, where it records the cells while migrating and proliferating. And with these techniques that I explained, we can conclude that inhibiting NHE3 gene expression in a short period of time causes colonic cells to proliferate and migrate more. So my remaining questions are, will the reduced expression of NHE3 lead to complications in the colonic cells, and can we inhibit NHE3 gene expression in the colon of an organism with IBD to repair its tissue?
Some personal questions I had this summer is, how can we use machinery and biotechnology to further scientific research, and how can we develop potential therapeutics for IBD patients?